Welcome back to the deep dive. Today we are peering right through the lens of molecular biology to uh, really understand this fundamental tension in nature,
Hmm.
the battle for control of the operating system. We are diving deep into how nature, using toxins, ancient parasites, and these highly evolved viruses, actively hacks, hijacks, and just rewrites the instructions of a host.
And it has a very specific recurring target.
The nervous system.
Exactly. This is, I mean, you could call it the ultimate biological espionage.
Mhm.
And for this deep dive, we're synthesizing a really fascinating collection of sources that show this control, it's not accidental.
Yeah.
It is an evolutionarily perfected strategy. We're going to move from the surgical precision of some newly discovered spider venoms all the way through the structural tricks used by viruses like rabies.
And even the dopamine hijacking capabilities of a parasite that's, well, it's globally prevalent.
Right. And then crucially, we're going to track where all this natural knowledge leads.
Hmm.
Our final segment confronts a purely theoretical but honestly chilling blueprint.
Project Chimera.
It's an extreme example of synthetic biology designed to merge all of these natural tools into a next-generation pathogen. And it's engineered for maximum genomic persistence and coordinated behavioral control.
The source material we have today is incredibly rich. We're covering the discovery of novel neurotoxins, the detailed structural biology that governs how well rabies vaccines work, the sophisticated genetic tools used to study parasitic brain manipulation, and of course that critical ethical and regulatory line.
Dual-use research of concern, DURC.
Our mission is clear. We want to provide you with the molecular blueprint for biological manipulation to understand not just what these agents do, but the foundational architecture that allows them to do it.
Because that same blueprint, whether you use it for good or for ill, is now driving cutting-edge synthetic genomics and, of course, biosecurity strategies.
Okay, so let's unpack this by starting with nature's molecular scalpels, the toxins.
Right. Animal venoms are, they're such this vast, concentrated chemical libraries. And they're often custom-built by evolution to target really specific neurological pathways in their prey.
But scientists, they see these compounds not as weapons, but as this invaluable toolkit for drug discovery.
Exactly. Our sources highlighted a search that involved screening 30 different animal venoms against one very, very specific human target.
The HKV 1.5 ion channel.
Right. And this ion channel is critical because it controls the flow of potassium out of cells, and it's a major therapeutic target for treating atrial fibrillation, AF.
A really common type of heart rhythm disorder.
Finding a compound that selectively modulates HKV 1.5 without, messing with other heart channels would be a massive breakthrough in AF treatment.
And in this particular hunt, one venom stood out.
The whole venom of a spider, Oculocosa supramirabilis, it just lit up immediately as a potent regulator of that channel.
And it's important to remember the dual nature here. The venom works on the human channel, which makes it medically relevant, but in its natural context, it's also a highly effective insecticide.
Yeah, against things like house crickets. So the researchers then had this big challenge. They had to chemically separate the whole venom to find the one single active compound responsible.
So they use a technique, HPLC reverse phase fractionation, to zero in on this active peptide, which they named OSU1.
But this is where the real technical challenge begins. Because isolating a tiny, potent peptide from a natural source, you get a handful of molecules.
Not enough to work with.
Not at all. To analyze its structure and its potential therapeutic function, you need much larger quantities.
Yeah.
And you need it to be pure.
And that's where synthetic biology takes over.
Yeah.
They decided to create a recombinant version, ROSU1.
Right. And the first step was constructing the gene de novo, which means they took the amino acid sequence they read from the peptide and worked backward to synthesize the DNA code that would create it.
Then they put that DNA into bacteria to express the protein.
But, producing these complex natural toxins is notoriously difficult. It's not like making simple bacterial proteins.
Right. These complex peptides, especially from something like an arachnid, they rely on incredibly precise folding and linkages to actually work.
If it folds wrong, it's just inert. It's protein sludge.
So that's the core challenge then.
It is. They had to purify that raw protein sludge, which often forms these insoluble clumps inside the bacteria. And then came the crucial step, the in vitro refolding.
Which is basically a complex chemical bath.
Exactly. It's designed to guide the molecule into its perfect functional three-dimensional shape. It's like, trying to perfectly fold a microscopic piece of origami.
Where the whole structure is stabilized by 4 chemical welds, the disulfide bonds.
Yes, and to make sure it folded correctly, They first had to chemically break any incorrect bonds that had already formed. Then they had to allow it to slowly reoxidize.
And this took a while.
Over a period of 4 days, in a very, very specific chemical environment. And that 4-day wait, just to ensure this tiny molecular structure sets just right, it really underscores how difficult it is to handle these natural weapons.
So once it was functional, did the recombinant OSU1 act like the native peptide?
It did. It showed comparable biological effects, including similar insecticidal activity against the crickets. They tested its potency by measuring the median paralyzing dose, the PD50, and the lethal dose, the LD50.
And what about in mammals? Did they check for safety?
They did. They checked for early safety markers, injecting it directly into the brains of mice, and they found it was non-toxic up to 5 micrograms per mouse.
Wow. So that selectivity, potent against insects and specific ion channels, but low acute neurotoxicity in mice. That's exactly what makes it such a valuable lead compound.
But the real scientific prize here isn't just its function, it's its structure.
Okay.
OSU1 has a hypothesized disulfide pairing motif. So the arrangement of those 4 stabilizing chemical welds, that is highly uncommon.
So the way it's folded is unique.
Very unique. It connects the cysteine residues in an arrangement cis28 to cis40, cis10 to cis26, and so on, that is just not seen in existing spider peptide classifications.
And did they model this?
Yeah, they used multiple sophisticated computational models, Modeler, ITASR, and Swiss model, and all 3 of them confirm this unique 3-dimensional structure. It's got a small alpha helix and a double antiparallel strand.
So the fact that its folded pattern is fundamentally different from other known spider toxins suggests what?
It suggests OSU1 represents a whole new family of spider toxins.
That's incredible.
What's fascinating about this is twofold. First, it just shows that nature still holds these immense libraries of biologically active compounds we haven't even cataloged yet.
And second?
Second, these precise, channel-specific toxins are exactly the kind of refined molecular tools that synthetic biologists are looking to leverage. They offer these highly specific ways to manipulate electrical signaling.
So when we talk about engineering nervous systems later on in the show,
And these peptides are the keys.
that structural precision is just astonishing. Okay, now let's shift from a single fast-acting peptide to an entire organism that executes this complex, slow-burn behavioral hack.
Right.
We're talking about Toxoplasma gondii, a parasite whose whole strategy involves completely rewriting the risk assessment of its host.
T. gondii is, it's highly prevalent globally. Estimates suggest billions of people are infected, but its life cycle is very specific.
It needs a cat.
It absolutely requires a cat or any member of the feline family as its definitive host,
Right.
because that's the only place where it can complete its sexual cycle and shed infectious oocysts.
And we, humans, like rodents, are just intermediate hosts. We get infected, but the cycle, it stops short of transmission.
And when we're infected, the parasite forms these incredibly stealthy structures called tissue cysts.
Right.
And these cysts can contain up to 3,000 parasites. They can get up to 200 micrometers in size. They can form anywhere, the retina, muscle tissue, but they are most commonly found in the brain.
And the true masterstroke of this parasite is that these cysts can remain in situ for the host's entire lifetime.
Without provoking an immediate inflammatory reaction from the surrounding tissue. They are, for all intents and purposes, ghosts in the machine.
And this quiet persistence is directly related to its core survival strategy, which is host manipulation.
To complete its cycle, it needs its intermediate host, usually a rodent, to be eaten by a cat.
And this is where the behavioral hack comes in. Infected rodents, they don't just become, generally sick or sluggish.
No, it's highly specific. They exhibit a distinct change. They lose their innate, genetically hardwired aversion to cat odors.
In some cases, they're even attracted to the smell.
Exactly. It's a targeted form of, you could say, suicidal recklessness, designed solely to increase the probability of predation.
This is one of the clearest examples in nature of an organism actively controlling the mind of another for its own propagation. But how? How is this small parasite causing such a dramatic shift in complex fear processing?
The research points overwhelmingly to one thing: altered neurotransmitter signal transduction.
Specifically dopamine.
Specifically dopamine metabolism. When they compared infected rodents to uninfected controls, the infected group showed a significant increase in global dopamine content in the brain, about 114% of the levels found in uninfected mice.
Wait. So the parasite isn't just irritating the host brain, it's actively producing the neurochemical needed to drive this reckless behavior. That is profound.
It is. The parasite is carrying the necessary tools. The studies found that T. gondii possesses the gene for tyrosine hydroxylase.
Which is the key rate-limiting enzyme for dopamine synthesis in mammals.
Right. And that gene is encoded right there in the parasite's own genome.
So the parasite becomes its own little micro-dopamine factory.
Precisely. And they confirm this by looking at how the parasite behaves both in vitro and in vivo. When researchers infected dopaminergic PC12 cells, they saw that the infection enhanced the level of potassium-induced dopamine release by several folds.
And this enhancement was directly correlated with the number of infected cells present.
It was. And in the brain tissue itself. In chronically infected mice, when they stained for dopamine, the staining was intense and was concentrated primarily within the T. gondii tissue cysts.
Not just in the host neuron.
No, in the cyst. Specifically, it was punctate and localized near the periphery of the cyst wall. The parasite is essentially building and maintaining its own neurochemical manipulation station right inside the host's brain cells.
That is just a masterstroke of parasitic manipulation. It's creating the precise neurochemical environment it needs to facilitate its own transmission. But we as humans, we're dead-end intermediate hosts. What are the implications for us, considering this deep connection to dopamine dysregulation?
Well, the human implications are significant. Dopamine dysregulation is a central feature in many psychobehavioral conditions, most notably schizophrenia.
And research has found links.
Consistently. Research identifies associations between T. gondii seroprevalence, which just means a person has been exposed to the parasite, and an increased incidence of schizophrenia.
And well, correlation is never causality.
Right. But the fact that the parasite actively modifies the host's dopamine system, a system centrally implicated in schizophrenia pathology, that certainly gives those epidemiological associations strong biological plausibility. It warrants intense research scrutiny.
To do this kind of intimate research, scientists need incredibly robust genetic tools.
They do. To study and manipulate the parasite genome, especially for the highly virulent Rh strain, they use this clever technique called phosmid recombinaring.
Can you simplify what that tool allows them to do?
Think of it like a highly stable, controllable storage system for massive chunks of DNA. The phosmid is a type of vector that is maintained as a single copy per bacterial cell during normal storage.
Which is crucial for stability.
Exactly. When you're dealing with large, complex pieces of DNA, that's key.
But then, when they need bulk DNA to work with in the lab, say, to transfect the parasite cells, they need millions of copies.
Right. So the same vector has this clever switch. They can introduce an inducer molecule that triggers a second high copy number origin of replication. It lets them store it safely as a blueprint, and then, on demand, hit a switch to mass manufacture the DNA they need for high-throughput experiments.
And this precision allowed researchers, for instance, to rapidly pinpoint a single-point mutation V728G in the TGRCC1 gene that was responsible for a specific temperature-sensitive growth defect in the parasite.
Right. These tools are the foundation of our current understanding. They show us how these tiny molecular changes translate into complex behavioral outcomes. And they're also the very foundation that Project Chimera-L is built upon.
We've seen the slow, strategic persistence of T. gondii. Now let's move to the acute viral terror of rabies. This CNS infection, caused by a Lyssavirus, is defined by its near-perfect lethality once symptoms appear. The fact that fewer than 10 human cures have ever been medically reported worldwide, that tells you everything you need to know about its efficiency.
Its lethality is virtually guaranteed. The process starts when the virus is transmitted through saliva, often via a bite, from an infected mammal. It replicates locally in the skeletal muscle tissues near the entry site, and then it begins its journey.
The slow march to the brain.
Right. It travels along the nerve pathways via retrograde axonal transport, slowly making its way to the central nervous system.
But I guess you could say the genius of the virus is that it actively engineers the final stage of its life cycle. Transmission. It directly targets communication pathways to induce the aggressive, frenzied behavior that makes the host bite and spread the virus.
And the molecular key to this frenzy is the rabies glycoprotein, or G protein, which studs the surface of the virus. This G protein is structurally critical for two things.
Okay, what are they?
First, allowing the virus to get into the muscle cells initially, and second, interfering with receptors in the brain later on.
And what interference does it cause?
Well, research has shown that the rabies G protein contains a sequence of amino acids that's almost identical to a known sequence found in certain snake venoms.
Snake venoms again, so it's another neurotoxin.
Essentially. And this sequence is specifically designed to bind to and inhibit nicotinic acetylcholine receptors, NAC-ORs in the brain.
So if the innate killers are inhibited, how does that create the frenzied state?
These receptors are absolutely vital for communication throughout the nervous system. By binding to them and, jamming them, the G protein interferes with baseline brain function.
And that inhibition, coupled with the virus's broader neuropathogenesis involving interference with other key neurotransmitters like serotonin and GABA, creates this environment of chaotic communication failure.
Right. The result is that characteristic aggression and hypersensitivity. It induces the host to act out the behavior necessary for viral transmission.
Okay, shifting to countermeasures. The G protein is also the sole immunogen used in the rabies vaccine. But here, the structural science reveals a surprisingly subtle vulnerability in our defenses.
The efficacy of the vaccine is fundamentally tied to how that G protein is decorated with sugar chains.
And linked glycosylation.
Right. This is one of the most fascinating findings in vaccine science. Researchers compared two types of rabies vaccines grown in different production systems, one grown in viro cells, the VRV, and one grown in KMB17 human diploid cells, the HDCV.
And the protein sequences were identical, but the cell lines attached different sugar chains.
Exactly. And the differences were subtle, but they mattered so much.
How so?
The G protein structure is a trimer, 3 proteins bonded together, which is the structure the neutralizing antibodies must recognize. The HDCV G protein had sugar chains attached to 2 specific sites, but the VRVG protein had chains at all 3 sites.
And the extra sugar chain made a difference.
A huge difference. The additional sugar chain modification on VRV, specifically a hybrid glycan at amino acid position 319, forced the protein to form an excessive number of internal hydrogen bonds.
So instead of being dynamic and flexible, the sugar chains made the whole structure too rigid and unstable.
Exactly. Neutralizing antibodies need to see the protein in its dynamic native conformation. This excessive internal bonding in VRV increased the instability of the trimer structure. It essentially hid or distorted the antigenic features that the immune system needed to recognize.
And this instability is hypothesized to be the reason the VRV vaccine showed decreased protective efficacy compared to the HDCV-produced G protein.
Which maintained a more natural, dynamic conformation.
And the proof was in the experiment.
It was. They chemically stripped the sugar chain's deglycosylation from the VRV, and doing this significantly improved its immune protective efficacy.
Which is a remarkable validation. Removing the sugar coat that the host cell had accidentally added made the virus look more like its authentic, dangerous self to the immune system.
And conversely, deglycosylation of the HDCV had no such effect, which proves the VRV's issue was specifically related to the excessive stabilization caused by that third glycan site. It just shows that in structural biology, sometimes a biological shield meant for stability can actually undermine efficacy.
We mentioned the near 100% lethality of pathogenic rabies, but we also have research looking at what happens when neurons do survive.
Right. This research used an attenuated or weakened RABV strain in something called Cree reporter mice.
And this experiment was designed to challenge the old assumption that viral pathogenesis is simply about mass neuronal death.
Right. They gave the mice a sublethal dose and found that a significant proportion, 65 to 75%, survived the acute infection phase, and they used genetic markers, EGFP plus TERID, to tag the specific neurons that had been infected and survived.
And when they looked at these cured neurons months later, what did they find?
Long after the virus is cleared from the system, up to 3 months post-infection, the formerly infected neurons were not the same as their uninfected neighbors.
How are they different?
They exhibited permanent transcriptional differences, meaning the expression levels of certain genes were altered for good.
And what were these altered genes responsible for?
The changes were subtle but significant. The genes most affected were those crucial for cell-to-cell signaling and general nervous system function.
So what were the predicted consequences?
Bioinformatics analysis predicted functional consequences that amount to permanent damage. Specifically, decreased neurite growth, decreased organization of the cytoskeleton, and decreased microtubule dynamics.
If we translate that, it means the neuron is still alive, still there, but its ability to grow, organize its internal structure, and communicate effectively is permanently compromised.
Exactly. This supports the hypothesis that the functional damage of pathogenic RABV is due to neuronal dysfunction, not necessarily massive neuron loss. The virus keeps the neuron alive just long enough to promote its frenzied behavioral goal, and even when cured, the cell carries a permanent defect.
Which is a profound insight. Nature's goal might be functional damage and persistent system compromise rather than acute destruction.
And that observation leads us directly into the blueprint for a next-generation threat.
The last three sections have built a pretty compelling case that nature has perfected the art of precise neurological manipulation. Now, we confront the theoretical convergence of all this knowledge. Project Chimera-L.
An extreme example of what scientists categorize as dual-use research of concern.
DURC, it's the ethical fence line.
It is. It recognizes that research intended for humanitarian benefit, like developing aerosol delivery methods for vaccines or synthesizing potent toxins to target cancer cells, can easily be misapplied for malicious purposes.
And Project Chimera-L exists purely as a thought experiment, but it showcases the molecular architecture necessary to engineer a persistent biological control agent.
The theoretical aim here is, it's chilling, not simple, fast lethality, but engineered, persistent behavioral control and extreme transmission efficiency.
The goal is to create an undead plague carrier state.
Right. Using a rhabdovirus backbone, similar to rabies, but armed with 4 non-RABV components. Let's break down how this engineered synergy would work.
Okay, where do you start?
You need long-term stealth and host defense suppression first. This is achieved by incorporating Toxoplasma gondii effectors into the viral genome.
As we discussed, T. gondii is brilliant at suppressing host defenses,
specifically targeting things like miR-NAs and the NFKB pathway. So how does that translate into synergy within the Chimera-L framework?
It creates a chronic, immune-suppressed, fear-averse state.
Exactly. By suppressing early inflammation and cellular self-defense mechanisms, this agent ensures the infection can proceed chronically and quietly. The synergy creates what you just said, a transmission-maximizing carrier state. The host lives longer, carries a higher load, and has a dampened fear response, increasing the high-risk behavior needed for viral spread.
Okay, next, tackling the speed problem. Natural RABV is slow. It relies on that retrograde axonal transport.
So Chimera-L accelerates the process by incorporating leech factors, vasoactive peptides.
Derived from blood-feeding organisms.
Right, like leeches. Their function in this blueprint is to increase localized vascular permeability and deliberately compromise the integrity of the crucial blood-brain barrier, the BBB.
So by breaking down the BBB, the virus can bypass that slow, weeks-long axonal transport system.
Precisely. And the synergistic effect is massive. It allows a portion of the viral load to enter the central nervous system rapidly via systemic circulation. It accelerates the viral load increase in the CNS, maximizing the speed of symptom onset and the eventual frenzied attacks.
Component number 3 is the acute behavioral trigger, the so-called hive mind factor,
which relies on the precision of the toxins we discussed in Section 1. The blueprint calls for incorporating spider neurotoxins, paralleling the highly specific OSON.
And targeting synaptic ion channels.
Like voltage-gated sodium or calcium channels. These are the equivalent of molecular shock triggers.
Why this specific choice?
The synergy here is the immediate induction of acute, chaotic, synchronized neurological dysfunction, spasms, violent, frenzied attacks. And the timing and intensity of this trigger can be carefully controlled by which ion channels are targeted and how the toxin is expressed.
Ensuring maximum transmission efficiency just before the host expires, the hive mind effect.
Exactly. And finally, the element that makes this a long-term evolutionary nightmare, achieving genomic persistence.
Natural rabies is an RNA virus. It stays in the cytoplasm and is quickly cleared.
But Chimera-L incorporates a reverse transcriptase, RT enzyme. The RT enzyme allows the chimeric RNA genome to be transcribed backward into a DNA provirus. This DNA can then integrate into the host chromosome,
establishing the foundation for genomic persistence and long-term latency.
The theoretical reservoir of the undead plague carrier state. This fundamentally changes the nature of the disease from an acute infection to a chronic, potentially latent genomic threat.
And if that wasn't complicated enough for biosecurity experts, the blueprint introduces this counterintuitive feature called the volatility paradox.
Intentionally engineering instability into the genome.
That's truly advanced engineering.
It is. The plan calls for incorporating highly unusual left-handed DNA, or LDNA, segments into the genome. LDNA has a helical structure that twists in the opposite direction of standard right-handed, or ddNA.
So why would you deliberately include a chemical structure that your own replication machinery can't handle?
Because the viral RNA-dependent RNA polymerase, the rDRP, and the D-reverse transcriptase, the DRT, are evolved to handle standard ddNA. When they encounter the LDNA segments, the enzymes stall or terminate.
Ensuring a high error rate.
Exactly. A high error rate in the final synthesized product. which creates mutation. Add to this the fact that the RT enzyme naturally lacks proofreading, guaranteeing even more errors during reverse transcription.
And then there's the host's repair machinery.
Right. Once that highly mutated provirus DNA attempts to integrate, the LDNA segments are predicted to shield regions from the host's normal DNA repair machinery, specifically the enzyme DNA ligase IV, which is structured to recognize ddNA ends.
So the combination LDNA, no proofreading, and stalled replication guarantees genomic instability.
The result is that the chimeric pathogen doesn't exist as a single stable entity. It exists as a highly divergent quasi-species pool, a cloud of related but distinct genetic versions with inherent instability and accelerated mutation.
And chillingly, this intentional design compounds the difficulty of developing countermeasures because you are trying to vaccinate against a moving genetic target.
The entire concept of Chimera-L, especially the idea of using reverse transcriptase to switch the viral RNA genome into persistent DNA, it relies entirely on our ability to precisely control and engineer enzymatic function.
So let's look at the real-world research showing just how fine-tuned this control is.
We're looking at the Maloney-Murine leukemia virus, or MLMLV, reverse transcriptase, RT. The wild-type enzyme is naturally a DNA polymerase.
Its job is to synthesize a DNA strand using deoxyribonucleotides, DNTPs, as its building blocks.
And crucially, it absolutely rejects ribonucleotides, RNTPs, which contain an extra oxygen group.
And what's the physical barrier that prevents it from accepting the ribonucleotides?
Structural modeling provided the answer. The active site of the enzyme is very tight. A specific amino acid, phenylalanine 155, or Phe155, has this large, bulky side group that physically clashes, sterically hinders the incoming ribonucleotide because of its 2-OH group. The space is just too small.
So the researchers hypothesized that if they substituted that large amino acid with a much smaller one,
They could essentially open the active site door.
and that was the experiment.
Yes. They substituted the bulky Phe155 with the smaller vanillin residue, creating the mutant enzyme RTF155VH. And the experiment worked beautifully. The mutant enzyme gained the stunning capacity to incorporate RNTPs into products.
They had effectively conferred RNA polymerase activity to an enzyme that was strictly a DNA polymerase.
With a single amino acid substitutions, it's a fundamental shift in function.
What did the kinetic results tell us about the efficiency of this switch?
The change was dramatic. The mutant enzyme's chemical affinity, measured by its Kielman value for RUTP, the new building block, increased to be comparable to its affinity for DTTP, its natural building block.
Which strongly implies that Phe155 is the structural gatekeeper dictating what the enzyme chooses to build with.
It is. The enzyme became just as good at finding the new building block, even though it was still performing the synthesis function a bit slower.
How much slower?
The maximum speed, the Vmax, of synthesizing RNA products remained slower, about 100-fold less than for DNA products. But the enzyme was undeniably capable of synthesizing short RNA products up to at least 6 nucleotides long. This is proof that synthetic biology can fundamentally rewrite the rules of enzymatic function with atomic precision.
And this capability directly relates to the broader discussion of genetic vectors and regulatory risk, especially concerning our Chimera-L concept. We use naked DNA vectors constantly in therapeutics.
We do. DNA vectors are simple, stable, and easy to produce at scale. They are the backbone of many next-generation vaccines for complex diseases like HIV, hepatitis C, malaria.
Where they're designed to produce therapeutic proteins inside the host cell.
Right. But the concern, as raised by regulatory bodies like the Netherlands Commission on Genetic Modification, or COGEM, is the possibility of this naked DNA integrating into the host genome.
Which is a valid concern.
It is. For naked DNA to work, it must cross both the cell membrane and the nuclear membrane for expression. Once in the nucleus, interaction with the host genome is theoretically possible.
But COGEM's risk assessment found that the frequency is extremely low.
Extremely low. The risk of unwanted integration is likely far below the natural background rate of spontaneous gene inactivating mutations that occur in the human population every day.
So for most therapeutic naked DNA applications, the risk is minimal. But the regulatory oversight changes completely when you add those specific synthetic features we discussed earlier.
Yes. The regulatory trigger point, the factor that elevates the risk to the highest level of oversight, is whether the naked DNA sequence contains features that make it replication competent in mammalian cells.
Or if it contains sequences that promote efficient uptake by bacteria, or sequences that facilitate recombination with existing viruses to form entirely new infectious agents.
It's a difference between delivering a blueprint that the host merely reads and delivering a blueprint that contains the instructions, the tools, and the encouragement to copy itself and permanently edit the host's system.
And that is precisely the definition of the Chimera-L blueprint, an agent designed to actively promote genomic integration and replication, using tools like the engineered reverse transcriptase to make it stay permanent.
The technological capability is real, and the regulatory challenge lies in keeping that power focused strictly on therapeutic use.
We've concluded a truly deep dive today, traversing an incredible landscape of biological control mechanisms. We started with the molecular precision of the OS1 spider peptide.
Realizing that creating a functional biological tool requires 4 days of careful oxidation just to achieve its ion channel blocking shape.
We saw how an ancient tiny parasite, T. gondii, carries its own genetic payload tyrosine hydroxylase to manufacture dopamine directly within its tissue cysts in the brain,
Flipping the fear response of its host to maximize transmission risk, establishing a functional basis for those psychobehavioral changes.
We analyzed the structural vulnerabilities of the rabies virus, discovering that vaccine efficacy is this fragile dance. It hinges entirely on the G protein trimer maintaining its dynamic stability.
Right. If that protein is slightly too rigid because of misplaced sugar chains, the vaccine's efficacy is compromised. And we learned that even surviving neurons carry permanent functional damage long after the viral infection is cleared.
And we connected these natural architects of manipulation to the theoretical blueprint of Project Chimera-L.
A synthetic composite designed for genomic persistence via engineered reverse transcriptase, accelerated neural invasion via leech factors, immune suppression via T. gondii effectors, and acute behavioral control via spider neurotoxins. The power of synthetic biology to engineer biological volatility, not just stability, is now undeniable.
The fundamental takeaway here is the acceleration of convergence. The high-resolution knowledge we gain from studying nature's molecular architects, the specific folding of a new toxin, the viral sequence used to jam a neural receptor, or the parasitic genetic toolkit.
It's immediately absorbed by synthetic biology.
Immediately. The line between treating disease and designing a threat is becoming increasingly blurred, and it's centered on fundamental tools like reverse transcriptase and our ability to generate genetically persistent, neurologically targeting agents.
And this acceleration, it requires a critical reassessment of our defense posture, which leads to a provocative thought for you to explore on your own time.
Okay.
If a pathogen's evolutionary goal shifts entirely from simple acute lethality, as we traditionally understand it, to establishing a state of long-term latency coupled with behavioral and cognitive control, as suggested by the attenuated RABV survivor study and the chimeral concept, how must our defense strategy fundamentally change?
Does our entire focus need to shift from treating acute infection and viral load to the far more difficult challenge of purging latent integrated genetic blueprints from the host genome?
And what does it mean for society when the single most powerful defense mechanism against biological threats, the rapid development of neutralizing drugs and vaccines, is intentionally circumvented by engineering genomic instability and forcing the target to exist as a constantly mutating quasi-species cloud?
The architect of the threat becomes the architect of the perpetual moving target.